Hello students, welcome back to the biology class. In the class we did the various types of genes or various definitions which can be given to the genes according to their characteristics or their functions. We did about the constitutive genes which are always functional and those which are non-constitutive genes which function only when the particular conditions are met, and they can be of two types, either inducible which are generally switched off and repressible which are generally switched on. We learned about the multi-gene family also, as well as the repeated genes and the single copy genes. The difference between the two is that repeated genes are those which are found in number of copies. Reason their product is very critical and essential. That's why they are present in a greater number to meet with the high demand of the product. And single copy genes are basically the 60 to 70% of the functional genes are single copy genes. We learned that pseudogenes have are developed because of mutation, because of which they fail to express themselves. So they are quite similar to the functional gene, but due to some error they are not able to function. Then we understood the concept of split gene, which was introduced by Roberts and Sharp, as well as the processed genes which developed because of reverse transcription or retroviral infection. We learned about the overlapping genes as well as the structural genes which encode for synthesis of proteins, and then the genes which regulate their functions are called as the regulatory genes, for example promoter, terminator, operator, as well as repressor. We learned about the jumping genes also in the last class. This concept was explained by McIntyre. Now the topic we started, we just had an introduction of the topic of translation in the last class, that it is the process of protein formation and it occurs in the cytoplasm when we talk about the eukaryotic machinery or system, because RNA formation occurs in the nucleus and then it undergoes various changes, and which are splicing, capping, tailing, followed by its transfer from the nucleus into the cytoplasm through the nuclear pores. And in the case of the prokaryotic machinery, it is always the cytoplasm where everything is going on, whether it is the replication, transcription or translation. That's why coupling of transcription and translation is only possible in the prokaryotic system, is not possible in the eukaryotic because it has to undergo changes. Now we learned about the machinery required for the translation in the last class, that the machinery involves ribosome, which are the site of protein formation, because translation itself is defined as protein synthesis. And then the raw material required for protein formation is the amino acids, and then the information, the various sequence of amino acids or the primary structure of protein is controlled by the DNA, which is the master copy. Master copy gives rise to the functional or working copy, and the working copy is the messenger RNA or the informational copy. So it acts, mRNA acts like a template for the formation of the protein, or you can say the sequence of the amino acids. tRNA is a transfer RNA which carries the amino acid according to the reading of the codon, because a long time back before your summer break we did the topic of types of RNA. If you remember, we have done that messenger RNA reading is done from 5' to 3', and the information present on it is called as codon. And the information present, or complementary information present on the tRNA is read from 3' to 5' direction, and it is called as the anticodon. So tRNA has a particular anticodon which will be complementary to the codon. Codon is the information of the messenger RNA. Then a very important enzyme which is called as the amino acyl tRNA synthetase enzyme, which catalyzes the complete process of the charging of the tRNA, etc. We learned about this diagram to some extent in the last class, that if you look at the RER, this is the lumen of the RER, and we all remember from 11th class that the ribosomes are attached in this manner to the RER. There is a hole present here through which the protein enters inside, or you can say the freshly prepared or nascent polypeptide enters from the ribosome into the lumen of the RER. Remember, in 11th we have learned about the proteins which are required for the attachment, ribophorins, and then remember that there are two subunits, the smaller subunit and the larger subunit. So the larger one makes a contact with the surface of the RER. Magnesium ions are very essential for this association of two subunits to form a functional ribosome, and because if the magnesium is absent from the scenario, then dissociation of these two subunits will occur and protein formation will stop or will not occur. You can see here there is a tunnel present inside. This tunnel is for the RNA to pass through between the two subunits, and then there is a groove present here. That groove is for the passage of the polypeptide, and it is present in the large subunit. Now from here the protein goes inside the lumen of RER. Now what are the reactive sites? There are 3 reactive sites. NCERT only mentions 2. NCERT says only P and A. So there are 2 reactive sites according to NCERT. I'll explain the third one also. So there are 3 reactive sites, P, A as well as the E. P is also called as the D site because D stands for donor and P stands for peptidyl transfer. Sorry for that mistake. Peptidyl transfer. A is for amino acyl or acceptor site, and E is for the exit site. Now the site P is synthesized with the help of both the subunits, but A and E sites are solely situated on the larger subunit of the ribosome. The enzyme involved is peptidyl transferase enzyme. We'll be learning about its function on the next pages. This enzyme is a ribozyme, means it is made up of RNA, and I have mentioned it here. NCERT only mentions about this as well as this. It does not give this name in the NCERT. So it's a part of the large subunit, and it is made up of 23S rRNA. Remember, this is the rRNA present in the larger subunit of prokaryotic ribosome. The smaller subunit has the recognizing site for the messenger RNA as well as binding area for the initiation factors.